plan for today is to finish up the last couple of concepts in the area of community ecology, and then go ahead and start ecosystem ecology. A couple of announcements, reminders. As usual, before any test, practice questions have been posted on EEE website in the usual spot on the grading and the test tab. And the instructor evaluation is still available just for the last few days. It closes Saturday night, so make sure you participate in that. You get 1% of the overall grade in the class for participating. Of course, final exams are next week. And your biosci peer tutors would like you to remember that they're holding a review session this coming Friday evening at 5 o'clock in PSLH 100. Okay. Well, the last few individuals are filing in. I'm going to go ahead and I don't like to do a clicker question right off the bat because I know some of you are running from the parking lot or the other side of campus, but I'll go ahead and start this up because I don't want to interrupt my So the faculty who are involved in teaching the core courses are asking their students a number of questions for evaluating the role of clickers and attendance in the classroom. So here's the clicker question. If clickers were not worth points, would you still regularly attend class? Yes or no? Please go ahead and click in. I'll share the responses in aggregate anonymously with the other instructors who are also asking their classes the same thing. Yes, those of you who are just coming on in, the question is, if clickers were not worth points, would you still regularly attend class? Please pick A for yes or B for no. And I'll close it down. Okay, everybody have a chance to click in? Alrighty, let's see. Let's see what you think. Not sure why you're groaning. So 2/3 would regularly attend class, so presumably 1/3 of those of you who are actually in the room would not. Okay, this is obviously just one question of a number, but I'm not going to take any more class time with this. But what I am going to spend class time with, because of some questions I've had from some individuals, which made me think that maybe there might be some need to spend a little more time on some of the population biology concepts, but I don't know. Let's try this one out. Let's do this as an any answer count. So this is one of the concepts from the population ecology, thinking about how to make calculations about birth and death rates and so on. Consider a population that has exactly 1,000 individuals in it, which is of course numerically very convenient. If the birth rate at the population in the current time is 40 new individuals that are born per year, what is the per capita birth rate per year right now in that same population of 1,000 when you're expecting 40 individuals to be born? Is it 40, 1,000 plus 40, 1,000 divided by 40, 40 divided by 1,000, 1,000 minus 40? What do you think? If you're not sure, chat with your neighbor. I'll give you 20 seconds or so to ponder this one. Go ahead and click in, do your best. This will help me evaluate how comfortable you are with this calculation. Okay, hopefully everybody's clicked in. Ready? Let's see what you guys think. Here? All right, well, 3/4 of you got it right. Absolutely. Okay, so those quarter of you that didn't, this would be a relatively easy entry-level question for thinking about how to deal with birth rates or death rates in a population. So depending on what tripped you up, this might be a good thing to spend a little time on in office hours or prep for the test. Specifically, what per capita means is, on average per individual. So to go from births to births per individual, right, where births, we learned at least the algebraic notation that we're using in this class is big B for just the straight number of births. So in this particular case, this is 40, right? And then capital N is the number of individuals in the population. And so at the current time, that's 1,000. The birth rate per capita is small b, right? So this is the birth rate per capita, where per capita means, per person, right? So that's the number of births divided by the number of individuals, right? Number of births divided by the total number of individuals, as most of you, of course, if you got that right, right? So, the same logic is used with going between total death rate and per capita death rate. Okay, so you should be comfortable going back and forth between total and per capita. But again, that's a good thing to try out in the review session or in office hours. We're having a hard time with that. Well, let's go back to the topic at hand, which is community ecology, and we'll consider community structure specifically. So community ecologists are interested in questions like, well, what species form a community? How many are they? How do they function together? And so community ecology, though, is much more than just making a list, going somewhere and just sort of writing them all down and going, well, here we are. Here's our list of species in the community. Community ecologists are very interested in the mechanisms, how many different kinds of species is possible for a particular area. How is the community respond to perturbations? So if one has a particular community, such as this picture right here from one of the familiar biomes that we discussed earlier, you can note that there are a number of different species that make up this assemblage, this community of living organisms. Well, what happens if one of these species is removed or something happens to it, it dies off? How does the rest of the community respond? Does it matter? Does another species move in? What's the relationship between the new species and the one that left? How are the other species impacted? So these are the kinds of questions that community ecologists are very interested in, and of course you can imagine these kinds of experiments can be quite tricky logistically to perform, but they're really important for making inferences about the way that communities actually function. There's one experiment on community composition that's described in your textbook, which I'll take a little time to discuss right now. So this is an example of how one can look at community, at least in initial formation, which is quite different than a manipulation on an existing community. This is figure 5515 in front of you right now and shows the kind of experiment that one can do if one is interested in, if a new habitat suddenly becomes available, like this, new pond, what kind of community will naturally develop within this new area? How predictable is it? So what the scientists did that did this experiment, they took an area and they made 12 ponds. These are artificial. These are not natural ponds. They're all exactly identical for themselves to each other. And they're filled with sterilized water. So you're starting with no living creatures of any kind in the water. Just an artificial pond full of water. That's it. Leave them for a year and then come back. So the community ecologists who did this were interested in, well, what happens? Do you end up with exactly the same thing in all 12 ponds? Or are they all completely different? What happens? So they did this experiment. They went back 1 year later and looked at their 12 ponds, and this is what the results look like. This is figure 5515 from your textbook, and this looks a little bit like an electrophoresis gel. It's not. It's just a way of graphing the results. So up at the top here we have ponds, and there are 12 different ponds, so each one of these columns is a separate individual pond, and then what the rows represent are different species. Now I'd also like to point out what species were they even looking at. The taxa that they actually evaluated was just the plankton. Okay, so these are not microbes. These are eukaryotic plankton, so things like algae, little zooplankton. So each one of these rows then, which is a different color, represents a different species. And so running your eye back and forth, you can see a couple of different patterns, right? You can see that there are a number of different species that were found in every single pond, like this one at the top, the red one, and then the greenish one, and the orangey one, and then the gray one, and so on, right? So there were some species that were certainly in every single pond, but as you roll your eye down the list here, you can start to see, because they're ordered so that it's a little easier to see the pattern, that there were certainly some species that were only in one pond or were not in other ponds. So what this shows then is that the results are somewhat between those two extremes. It's not that every pond is exactly the same, right? And so where did these plankton come from anyway, right? They were being seeded from the environment. The scientists did not add anything to these ponds. They just let whatever blow into the pond from the surrounding areas. So the ponds are not all completely distinct from each other, but neither are they all exactly the same. There were a total of 61 different species of plankton, so 61 different species of plankton. And yet when they counted up how many different species of plankton were in any given pond, the numbers are actually tabulated down here at the bottom. So pond number 1 had 35 species, pond number 2 had 31, 38, and so on. If you eyeball all these numbers, you'll notice they're actually in a pretty narrow range, right? Out of the 61 species that are in all of these ponds altogether, you don't see numbers like 2, right? There's no pond with just 2 species. There's no pond with 59, right? The number of species that occurred naturally or pseudo-naturally in these artificial ponds was actually fell in a certain narrow range. So this is the kind of experimental manipulation that community ecologists can do to try to understand some of the constraints or the boundaries within which communities operate. Another really important concept in community ecology is the idea that not every single species within a community necessarily is equally impactful as all the other species. So thinking about the plankton that we were just considering in the ponds, it's not necessarily true that every single species out of the, say, 35 or 38 or 31 species of plankton would necessarily be as impactful for the community as a whole, meaning if that species were to suddenly disappear from the assemblage. One example of a type of species within a community that is particularly impactful is the so-called keystone species. This particular term is given to a consumer specifically within a community. So it can be either a predator or an herbivore, something that eats meat or something that eats vegetation or something that eats both. So it's not a producer. But the species is not necessarily the most numerically abundant, nor is it necessarily the biggest individuals in the community. So you can't necessarily tell just from examining a community if there is a keystone species within this community, and if so, which species it is. One can identify or define a species as being a keystone species if it has a very major ecological role on the community. And one of the classic examples of this is a sea star, this handsome creature right here in front of you, figure 5516. Which is in genus Pisaster, shown right here. This is a voracious predator, so the sea star is in phylum Echinodermata, right? Remember that? And the kind of habitat that this particular sea star lives in looks like the picture here. There's a very diverse assemblage of organisms, other plants and animals and algae that live in the rocky intertidal areas that are favored by Pisaster. So a community ecologist was very interested in the role of the Pisaster, this particular consumer, within the community, and he decided to go to several areas in the rocky intertidal and try removing the Pisaster from some, but not others, and then follow these communities and see if there was any change, and if so, what was this change? The results are shown right here. In this graph, the x-axis is year. So down here in 1963 is when this experiment was started. This was a multi-year set of observations. And so the year when it was started, let's see, look at the y-axis here. The y-axis is numbers of species present. And so you can see when these experiments were begun, the areas that included the piezoester sea stars had on the order of 17 or 18 or so species within the community. And this is, again, not including microbes. This is just big macroscopic animals and plants and algae. After the piezoester were removed and the community ecologists went back and examined changes in the community composition, they noticed that there was a huge drop-off in the number of species when the piezoester was removed. And so it went from about 18 here to about what, two or three over the next several years. So a huge change in the diversity of species. Now this wasn't just an example of something that was going on in all the rocky intertidal areas in the region that had nothing to do with piezoester. And how can you tell? Because of the control. Here's the control line right here. So the neighboring areas where the piezoester was not removed maintained similar species diversity, even increased a little bit as shown here. So what's going on here? Well, these sea star preferentially eat mussels. Mussels are a kind of bivalve. This is something that humans eat too, so they may look familiar to you. But normally in the habitats when there is piezoester, they preferentially eat the mussels, and that keeps the mussel numbers down in the community. And so in the areas where there was no piezoester, then the mussels spread like crazy. They're very good at competing with the other organisms for space in the rocky intertidal, and so they would form these thick, dense mussel beds, crowding out the other competitors, driving down the species diversity to just 2 or 3 different species there. So a huge impact on these communities where they basically turned into mussel beds. So that's an example of a keystone species. Sadly, at the current time, this particular keystone species has been in the news a lot. Why has it been in the news? Well, there is something going on the entire West Coast, meaning of the United States, meaning Alaska to California, so including Canada as well, where this disease is called wasting syndrome. It's really not quite completely clear what's going on. It certainly seems to be associated with increases in the water temperature in these environments. Let's go ahead and go visit a website that is keeping track of this wasting disease. So some scientists at the University of California, Santa Cruz have set up a website where there's monitoring and information about the sea star wasting syndrome and maps and updates, and anybody who wants to get involved can submit observations here and collect data and so on. There's time-lapse movies. But basically what happens is that these sea stars, I'll scroll down a little bit, this first came to the notice of individuals who would go to the rocky intertidal, and instead of seeing normal 5-armed sea stars, would notice things like this, where there would maybe just be a single arm, or what looked like a little bit of a disintegrating sea star. And these are sea stars that have this wasting syndrome, and they can deteriorate extremely rapidly over just a day or two. So there are pictures down here. If I scroll down, the first symptom is usually some sort of little white patches that start to form, and then this spreads over the body. And then the individuals often start to lose arms. They disintegrate. So when one sees individuals with a smaller number of arms, that's usually why. And these white areas continue to grow and spread and so on. So they're affecting a number of different sea stars, not just this particular species, but some of the other relative ones as well. So anyway, there's where you can go if you're interested in more information on this disturbing syndrome. But considering the role that these sea stars do have in the communities that they're part of, then that is leading to additional concern, of course, of what's going to happen to these communities if the sea stars are going to be taken out by whatever's killing them. Then what's going to happen to the remaining communities? Are we going to lose a lot of species diversity? Okay, we'll now make the transition to the next more inclusive level of ecology. As you we're going through different kinds of ecology that are more and more inclusive, meaning include more factors. So the next step up from community ecology is ecosystems ecology, because an ecosystem includes both the community, the assemblage of species that make up the living organisms that occur together, but also all the abiotic components of the environment are also included when one talks about ecosystems. As mentioned before, community ecologists focus on interactions between species and also things like community composition, but ecosystems ecologists focus on flow of energy and also nutrients within ecosystems. So energy and nutrients. Different verbs are used to describe what's going on with energy versus what's going on with nutrients. So you see the term flow used for energy, and this is in contrast to the term cycle that is used for chemical elements. So why is this? Let's spend a few minutes talking about energy. It should be a familiar concept to you from the physical sciences. You learned a number of fundamental things about energy, presumably, which, for example, energy is conserved, it can be transformed. Energy within the context of an ecosystem is used in a slightly different context, although it's a very closely related subject. Organisms use up energy, and so there's a constant need for more energy than to be entering an ecosystem. So the verb flow is used to describe energy entering an ecosystem, moving through the different parts of an ecosystem, and being lost as organisms consume or use up energy. So this is in contrast to the actual nutrients or chemical elements, which are not used up. They can be transformed into a different chemical form, but they typically remain within the ecosystems, and we'll look at some examples of that later when we talk about cycles. But let's start by just considering energy flow within ecosystems. So as mentioned, organisms are using up energy, and so energy has to enter an ecosystem. How does it enter an ecosystem? Well, it enters an ecosystem at a level that is referred to as primary production. And in almost all ecosystems, this is the photosynthesizers that are taking light energy and converting it to chemical energy, meaning making organic molecules, making energy available to those of us who are heterotrophs, right? So these are obviously autotrophs then. There are a very small number of ecosystems on the planet where light is not the primary source of energy entering an ecosystem. These are ecosystems that do not have any access to light, so these are very rare. So for example, there are some deep sea communities where there is absolutely no light whatsoever because it's so far down, where there are chemical sources of energy that are used by the unusual living organisms that make up the communities at some deep sea, for some deep sea communities. But the vast majority of ecosystems, and particularly the ones that you're most familiar with, all start with energy coming in as light, which is converted by the autotrophs to chemical energy. Now, because that's the entry point to energy, then entering the ecosystem, that sets the limit then, because no more energy is made at other levels in the ecosystem, so that's what one has to work with then through the ecosystem. So what are these amounts? Let's walk through figure 56-1 from your textbook, which is now in front of you. So here's the light coming in, there's the sun, it's hitting the surface of the planet, and one can quantify the amount of energy that is hitting the planet's surface coming in as light energy. And so here's an example of the number. Do you have to know this number? Of course not. Anyway, here is the number, and if you look at the units, this is kilocalories. Kilocalorie is a unit of energy per square area per year, okay, because you need per time and also per area. Obviously, you might be sitting there thinking, well, surely this is dependent on latitude. Absolutely, right, in terms of how much energy is hitting different parts of the planet, and also season, and so on. But this is a sort of a global average. This is a representative number. So here's the amount of energy coming in. Then what? Now we have a photosynthesizer. And the photosynthesizer is going to capture some particular fraction of that energy and convert it to chemical energy in the form of organic molecules. Estimates for what percentage of the total amount of incoming energy is actually converted that is hitting the surface of the planet is about 0.8%. So this might strike you as a really, really tiny number. It's a relatively small fraction, right? So why is it not closer to, say, 50% or 90% or some other bigger number? Well, of course, not all light hitting the surface of the planet is necessarily hitting a photosynthesizer, right? And furthermore, not all wavelengths of light can be used in photosynthesis, so only a fraction of the incoming light is the right wavelength for photosynthesis. And photosynthesis is not completely 100% efficient either, so it can't, and not all light is able to actually reach the appropriate biochemical machinery within the photosynthesizers. So given all those factors, this is one of the reasons why this number is in fact so low. So, but this is then the starting point. This is how much energy is available to enter ecosystems globally as primary productivity. Now notice it says gross primary productivity, because this is how much is being used, turned into non-light energy. But of course these photosynthesizers have their own costs. They're living organisms. They're burning up ATP for their metabolism and growth. And so almost half of all of this energy is in fact used up by the organisms just as part of their metabolism. So about 1/2 then of all of the energy is actually locked up then into organic molecules, or as what's referred to as biomass then, right? So this leaf here would be an example of biomass, where some of this energy has been then converted into chemical energy in the form of tissues shown here. So about half. So this is where this difference between terms comes from, gross versus net, right? Hopefully you're familiar with that difference. Gross is an overall total, but sometimes depending on the context, one wants to take part of that out. So you go to the store, you buy a box of Cheerios or something, then it tells you what the net weight is, because if you're interested in buying some, you don't really care what the weight of the box is, you care the weight of the actual product. So there's a difference between net and gross weight as a consumer. So it is the net primary productivity then that is the amount of energy that is actually locked up in the photosynthetic tissues. So comparing gross versus net primary productivity then, and here are a couple of the acronyms in the chapter. Fortunately there aren't too many, so there's not too many to learn, but GPP is used for gross primary production and NPP is used for net primary production. So if GPP is the gross primary production, then this is how much light energy is being converted to chemical energy per area per unit time, and then you just simply subtract how much energy was used in the same period of time for the same amount of area, and you end up with a net primary production. I wanted to say a comment here about units. It should make sense to you, if we're talking about energy, that the best units might be things like joules or kilocalories, or some kind of unit that actually is an energetic unit, right? But you'll find that energy units are used, but also ecosystems ecologists often sometimes flip back and forth between considering energy units and also mass units, and you'll certainly see this in your textbook as well. So don't get confused by that. It's a bit of a shorthand going back and forth because if the energy is locked up in terms of of tissue, then you can actually weigh the tissue and convert back to the amount of energy that would be liberated if one were to consume it. So there is a conversion between those two, but ecosystems ecologists sometimes describe productivity in terms of mass per unit time per unit area, or mass per unit time per unit area, or energy per unit time per unit area. So don't find that confusing. They're both considered fairly proper units to use. So this is figure 56.2 from your textbook, which makes diagrammatically a nice comparison for the conceptual point about the difference between flow and cycling. So we've got energy flow on the left and cycling going on the right. So what is the way of thinking about this? Here we have the incoming light energy, so light energy entering the ecosystem here. The individuals that are photosynthesizing and converting it then are the primary producers, and you should be thinking to yourselves, yes, these are the autotrophs, right? The primary producers are losing some energy, as mentioned before. That's indicated diagrammatically here by the little wiggly yellow arrow here. These photosynthesizing individuals can be consumed by the organisms that eat them, right? So what is this? This is then obviously the difference between the autotrophs and the heterotrophs. Something that eats an autotroph and uses the chemical energy from the organic molecules then as a source of energy instead of light is a heterotroph, right? A primary consumer that eats vegetation would be what? An herbivore. So we're talking about an herbivore here. And then something can eat that herbivore. We've got a secondary consumer, it'll still be a heterotroph, but if it's eating an animal that ate some vegetation, then we're now talking about what? A carnivore, right? So you should be pretty comfortable with moving through, keeping track of who's eating whom and what the names refer to as you're considering these ecosystems. So notice that at every single layer here, we've got little yellow squiggly arrows indicating that all organisms are using up energy, and so that's energy that is being lost from the ecosystem. That's in contrast to what's going on the right here, where the nutrients are being recycled through the ecosystem, as indicated here with these different arrows. And I also want to point out a really important level here within the ecosystem, which are the so-called decomposers, which is a special name given to those living organisms that break down tissues, usually dead tissue, whether the dead tissue is part of a primary producer or primary consumer or whatever. Okay. So as we consider then the movement of energy between different individuals who are making energy and then consuming energy, then this is sometimes described as a trophic cascade, and this is diagrammed in figure 56.3 from your textbook, which is in front of you right now, starting right here with the primary producers. And one of the things I like about this particular figure is that it starts with living material and an example over on the left here. So we have an example of a live leaf here, and then we have a dead leaf over here on the right. So both of these would be considered examples of primary producers. Obviously, the production was done before that leaf died, but nonetheless, we did have conversion of light energy into the energy that's now locked up in these leaves that are alive. These leaves can then be eaten by consumers. Here they are. So we've got a cricket eating a live leaf, and we've got some microbes decomposing the maple leaves. Then we have something that can eat those in turn. So we're going up different levels, different trophic levels. Trophic means having to do with eating. And then notice that the names change as one goes up the trophic levels. We went from primary producer, there is no secondary producer here, right? We then go to a primary consumer or a primary decomposer. We now have a secondary consumer, which is the name given to an individual that's eating a primary consumer. Then we've got tertiary. Tertiary means 3. Quaternary, right? 4. Notice it stops here. So why did it stop? Did they just kind of run out of room, or there's not any more levels? Well, typically it's extremely rare to have ecosystems where one in fact does have more than 5, 6, or maybe at a very maximum 7 different distinct trophic levels. And we'll get back to that point in just a minute. But before I leave this particular diagram, hopefully you're noticing a couple of things about this, namely that, well, gosh, isn't that actually the same bird there? So a particular species can have a number of different roles within an ecosystem, functioning both as a secondary consumer and as a tertiary consumer, depending on what exactly it's eating. Same thing with this hawk here. And so, in fact, these kinds of different trophic levels are not always usually shown as a single ladder here, but usually diagrammed as a food web. And that's shown here, which is figure 56.4 in front of you right now, where now the various interactions between these organisms are shown. And these arrows indicate who is eating whom, where the arrow shows the direction of the food. So the robin is eating both the millipedes and the crickets and the pillbugs and the earthworms and so on. So it shows the relationship between these, and this is a somewhat more informative way of looking at this. But again, there's still typically usually 5 or fewer, with very few exceptions, between the producers here at the very first trophic level, the primary producers, and the very, very top predators who are eating the consumers, who are eating other consumers, who are eating other consumers. So why is that? Well, one way of understanding that is to recognize what's going on with the energy transfer between these different trophic levels. This is fairly similar to figure 56.6 from your textbook. It's actually not exactly the same, but it's fairly similar. What it shows is the different trophic levels and the amount of energy that is typically transferred between. So if we start at the bottom here, for every million joules of sunlight, so here this is a unit of energy, maybe about 10,000 joules out of that million, more or less, is then converted by the primary producers to chemical energy. This is then available to the next level. So this level here is this level of energy transfer is not 10%, but that's not between trophic levels, right? Sunlight is not considered a trophic level. Once you hit the primary producers, then if one considers for every 10,000 of joules of primary producers that is consumed by primary consumers, how much of that energy then ends up as part of the mass of the primary consumers? Well, it's only about 10%, about 1,000 joules. Same with the next level, down to about 100, tertiary about 10. So notice that as we get to higher and higher predator levels, there's less and less and less energy available at each level. And of course, the higher and higher predator levels are often, large, active, voracious eaters. These are expensive organisms, and it takes a lot of food to keep those predators alive. So one sort of runs out of energy available to support top, large, calorically intensive predators at the top of the food chain fairly quickly, losing 10% each step. So that's one of the reasons that there is not usually more than that many levels. So what's different from this figure, your actual figure 56.6, the next time you're looking at your textbook, look at the units there. That one's actually done in mass. I believe it's grams per something, right? So getting back to the point I was making earlier about ecosystem ecologists sometimes use energy terms and sometimes mass terms, but that's considered perfectly acceptable. And the numbers will be slightly different. 10% is an estimate. It can be slightly more or slightly less. To connect back to an important conceptual topic that was covered in the population ecology of the course, think back to comments that were made about the logistic growth equation in populations and how for a given level of resources available, then a population will hit some sort of carrying capacity based on resources available and resources consumed. So one of the reasons why is such a wide range in estimates about the carrying capacity for human beings on the planet Earth are caused by different assumptions in the diet. And this should make sense to you in the context of considering how much energy is available at the different levels at the different trophic levels here. So for example, the farther down in the food chain one is consuming, the more energy there is at that level for consumption. And so there are differences in calories available if one is consuming higher level predators versus lower level predators or vegetation. So if one assumes, for example, that humans are only going to be eating plant material, then one ends up with a different kind of estimate for the total population that can be sustained on the planet than if one assumes that humans are eating meat. So that's connecting up with that topic for you. So ecosystem ecologists focus heavily on net primary production for a number of different reasons. Compared to gross primary productivity, of course, the value here is that that's what's available to consumers. That's the starting point of energy. And even though much is lost as one passes to each trophic level, this is sort of a starting point which allows one to wrap one's mind around exactly how much biomass can we even support here. Not all ecosystems have the same level of productivity. And so the most productive ecosystems per unit area are tropical wet forests and estuaries and coral reefs. These are some of the most productive in terms of the amount of energy per unit time, per unit area that is being converted to chemical energy. Overall, the oceans are not terribly productive per unit area, but because so much of the planet is in fact covered by ocean, there's such a huge area that the marine ecosystems contribute a lot to overall global net primary productivity. One can get a nice feel for the global pattern of net primary productivity by looking at a figure such as this one. This is figure 56.9 from your textbook, which shows the net primary production globally as sort of a color diagram here, where black and sort of purple are the lowest levels of net primary production, and the closer you are up to orange and red, the higher the level of net primary production. And you can notice a couple of things looking at the globe. One thing, as pointed out before, the oceans are typically levels of per unit area, relatively low net primary production, but again, it's such a huge area that globally it is a very important source of energy input into ecosystems. Another thing to notice is that there's a particularly high level of net primary production in the tropics. So if you look here in the equator, up and down a little bit, the most intense areas of orange and yellow and so on are typically in the tropical areas, and this should not be a huge surprise because after all, the tropical latitudes are where the most intense sun is that's coming in and available to the photosynthesizers, and it's also where the temperatures are warmer on average. As one goes in the direction of either pole, then typically net primary production falls off. So what are the limitations to primary production? In aquatic ecosystems, whether we're talking about freshwater or oceans, light, naturally enough, is one of the main things that controls primary production, and so that means that in aquatic ecosystems, almost all of the net primary production is taking place where? In the photic zone, that top upper level within the aquatic ecosystems where light penetrates enough to still have photosynthesis possible. Right below the photic zone, there's not enough light there to support photosynthesis, and so with the exception of just the very small rare deep sea communities, then there's no primary production going on. But in addition to light, there are some nutrients that are often rate limiting for primary production, and the two most common are nitrogen and also phosphorus. So a variety of different chemical compounds that contain nitrogen or phosphorus are the most common limiting nutrients in aquatic ecosystems. If we compare that to what limits primary production in terrestrial ecosystems, in terrestrial ecosystems, it's also light, not surprisingly, but also moisture or water availability, rainfall. That's really important to terrestrial ecosystems, unlike aquatic ecosystems, because of course aquatic ecosystems, you've got water right there. Temperature is very important, and so are nutrients. And it's the same primary limiting nutrients controlling primary production in terrestrial ecosystems than aquatic. So really, there's more similarities than differences between aquatic and terrestrial ecosystem limitations in primary production. So this is actually where I left off at 1 o'clock. I know I've got about 3 minutes left. I'm going to go ahead and let you go. Maybe so I'll see some of you in office hours tomorrow at 2 o'clock. Otherwise, I'll see you on Friday.